subject, and hopefully you pick up a few important factual information that can help in management of these viruses. So looking at the overview of what we will go through today, I'm going to talk about some of the common viruses and a few of their characteristics. I will talk a little bit more in detail around respiratory syncytial virus and influenza. I will go through a bit of our statistics as part of AMPATH lab surveillance, and this speaks to the epidemiology, the prevalence of the various viruses that we see over the season. I will note a few important facts on diagnostic testing, and I'll conclude the talk within the hour. So to get going, and I think it's no surprise that we are almost into spring, but winter is still lingering on, and I'm sure you all have experienced the common cold or influenza, or you've had a family member or friend who's been through this over the past few months. So the viral pathogens certainly keep our practices busy during the season. Of note, the common ones that we find that cause upper respiratory tract infections are your rhinovirus, enterovirus, your RSV and influenza, which tend to be the main ones circulating. And we will also talk through some of the other common cold viruses in this talk. The important thing to note as part of your epidemiology is that the common cold may occur at any time of the year, but typically it has a high prevalence during the autumn and winter months here. In South Africa, we experience it generally between March into September. Your rhinoviruses, which have more than 100 serotypes, cause up to 50% of colds in both children and adults. And we also note an increase in your RSV and influenza virus and your common cold coronavirus infections. Note not your SARS-CoV-2, but your circulating human coronavirus infections over this period. The generalized incubation time, which is time between contact with infectious material until the onset of symptoms, for most of these viruses is between a day to 3 days. And what we know, this tends to be really common knowledge, but a few important things to note is that viruses may remain viable on skin for as long as 2 hours and can survive for up to a day on your inanimate surfaces. Porous materials such as tissues and cotton handkerchiefs do not appear to support virus survival, so that's a good thing. The risk of person-to-person transfer is also dependent upon the amount of time people spend with each other, the proximity of, how they are, how close contact they are with one another, and the amount of virus that is shed from the infected patient. And this generally translates into the viral load. Transmission is through droplet spread, and we normally break this up into a small particle droplet or a large particle droplet, and that is studied under the electron microscope, and that denotes to a small particle being under 5 microns. So inhalation of small particle droplets, otherwise known as droplet nuclei or aerosols, that become airborne from breathing, talking, singing, coughing, or sneezing is one way to transmit it. And then when you have deposition of your large particle droplets, which are greater than 5, those are expelled and they travel and land on inanimate surfaces, or they land and you inhale them in through your nasal or conjunctival mucosa, or also through hand contact where you're self-inoculating with virus. I think we all know too well about the times of COVID, and this was, extensively covered at that time. I think the important thing to note, and, there isn't a specific symptom that denotes a particular virus. So the symptom profile is shared. And the usual symptomatology is the congestion, the runny nose, the cough, the sneezing, and the fever, which could mean anything really. And this is just an illustration of that, where they looked at healthy school children, and this was found to be the common symptoms. So going further into what we see with our common cold viruses, and as I mentioned, rhinovirus and respiratory enteroviruses are the major cause of your upper respiratory tract infections. So they are classified as part of the genus enterovirus, and not to go into detailed virology, and it can be quite complicated, but just to know there are those that have a predilection for your respiratory epithelium, and they denoted as rhinoenteroviruses. Due to their numerous different serotypes and the fact that they have a high mutation rate, which speaks to their fitness and their evolution or their genetic diversity, there isn't any efficient antiviral treatment or vaccines against these very prevalent pathogens. So we don't really have effective management other than for mild self-limiting disease, which is just the usual over-the-counter medications that we would use. And as mentioned, they are benign, mainly self-limited, and you get over them. However, there are a few of them that can cause severe pneumonia, and these tend to be in your high-risk patient groups like your elderly, your immunocompromised, your very young, and they can cause exacerbations of your chronic obstructive airway disease and asthma. Of note, there are 7 human species. So rhinovirus tends to be specific to humans. We don't see this in animals. So there are 7 human species. Of note, there are 3 rhinovirus species that are subtyped A to C, and there are 4 enterovirus species subtyped A to D that tend to have a predilection for your respiratory tract infections. Although they are closely related at a genetic level, when studied by their phenotype under electron microscopes and in research labs, they are actually quite remarkably different. So tropism, as I mentioned, tends to be mainly upper respiratory airways, but in rare cases, they can cause extrapulmonary manifestations, and literature speaks of cases of gastroenteritis and some cases of pericarditis. Mainly with the wide range of respiratory illnesses, you'll find asymptomatic infection to mainly your bronchitis, bronchiolitis, and wheezing or pneumonia. Children are the main reservoirs of rhinoviruses. They can experience up to 8 to 12 infections per year, where adults seem to have it less frequently. And as mentioned, your incubation can be anything from a day to 2 days, but your symptom duration can go as far as 10 days. So the important thing in recent studies, speaking of the rhinovirus type C, demonstrated a different trend with a peak of infection during winter months. And this particular strain does tend to be slightly more virulent from what literature says. So on the point of immunity, the interesting thing is that your rhinoviruses, your influenza viruses, your adeno and enteroviruses tend to produce lasting immunity. But the immunity does little to prevent subsequent colds or infections because, as I mentioned, you have so many serotypes. So it would speak to the reinfections that we experience, and it's like, oh, I've got the cold again, or I've got the flu again. So it's the multiple reinfections from different serotypes that can occur in generally more, I would say, patients with high risk or factors for that versus your immunocompetent individuals. So the antibody response to your rhinovirus infection occurs with the development of serotype-specific neutralizing antibodies. And I think that's important. You want to kill off the virus, so your body produces antibodies that's going to neutralize virus. But you also get secretory antibodies that are produced at the level of the mucosa and the airways. And we normally detect this generally about a week or two after infection. And as mentioned, immunity can last up to a year. Generally, serological testing is not done, and this is based on academic papers and research from academia. Important to note that there is no cross-protection between the different serotypes, so hence also a challenge for vaccine development. With regards to your rhinovirus and your parainfluenzas and your common cold coronaviruses, those are of importance in that they do not produce lasting immunity. So there's a slight variation in that. I speak to the parainfluenza virus because I'm sure in some of your patient reports, when you do PCR testing, you would see patients come up with parainfluenza and one of the different types, and you think, what's the difference between the parainfluenza and influenza? But really speaking, they are all RNA viruses, and they just, cause similar symptomatology and the predilection for the upper airway or the respiratory epithelium of the upper airway. And with parainfluenza, again, you have different serotypes. They tend to cause disease in children that are younger than 5 years of age. The immunocompromised children will always be at risk for severe infection from any type of virus. Importantly to note, when you get reports, you'll see either parainfluenza 1 comes up, or 4 or 2. The interesting to note is that 1 and 2 tend to be associated with croup, whilst 3 tends to have more serious pneumonia and bronchiolitis. And your serotype 4, is generally both upper and lower respiratory tract illness. So it's just important things to note that may not really make much of a difference, but it may explain the clinical manifestations that you are seeing. So as mentioned, your natural immunity is incomplete and you can get reinfection with virus. Another important etiological virus is the human metapneumovirus, and these names can be a mouthful. But of note, because of more recent molecular diagnostics and the fact that you have great ways of now identifying viruses, this one was discovered in 2001, but it has been circulating for many more years prior to the fact that a PCR detected it. So it has a worldwide distribution, tends to be associated with respiratory disease, mainly in children. And your circulating virus strains exhibit minor genetic diversity, but there is no evidence to date that strains vary significantly in their virulence. So that's a good thing. And here it's transmitted through large droplets, again, can cause upper and lower tract disease, but it's self-limiting and mild in the majority of cases. Speaking about your common coronaviruses, I think this is important because it must not be confused between SARS-CoV-2 or your SARS-CoV, because these tend to be questions that we get called for. Is there a new coronavirus that is circulating when a respiratory PCR result goes out and you see one of these different serotypes that are reported? It's important to take away that these are part of the human common cold coronaviruses that circulate during our season, and it causes 1/3 of your community-acquired upper respiratory tract infections. So again, always, as I mentioned, there will always be severity in your respiratory infections in both adult and children based on the risk factors that they may have. From an immunocompromised perspective, chronic kidney disease, prematurity, or whatever, the list can be exhaustive, but just to know. So of note here, there are 4 genera. The human coronaviruses are in 2 of these genera, the alpha coronaviruses and the beta coronaviruses. So your 229E and NL63 fall under the alpha, whereas your HKU1 and your OC43 are found under your beta coronaviruses. And we tend to send this out in our reports as part of the assays that are tested. Also, you find that your MERS-CoV falls under your beta coronavirus, and your SARS-CoV and your SARS-CoV are also in that beta coronavirus. These are all associated, these 7 coronaviruses are associated with human disease. Now, speaking a bit on viruses and wheezing, and I think it is an important thing to touch on, because in your practice during this time, you will always find, or more often rather than not, patients complain of a tight chest and the query of, do I have asthma or what's going on, or the absolute concern that they have a symptom that they've never had before. So the interplay between viruses and wheezing is a real one, and in your busy practice, I am sure this is something that you experience regularly. So they are a common trigger of wheezing illness in children, and your virus-induced wheezing represents a heterogeneous group of illnesses where you could have bronchiolitis, asthma exacerbations, and croup. So as mentioned, your respiratory infections have a tropism for your airway epithelium, and they interact with host factors to promote an inflammatory response. And that inflammatory response actually triggers the wheezing. So you can have an exacerbation of that may give you a more severe symptomatology, or it's the interplay between virus and host that actually speaks to what goes on at the cellular level. In susceptible individuals, the virus-induced immune response may mediate host responses to other microbes as well, infections, to allergens, to stress and pollutants, and this also results in wheezing and exacerbations of asthma. Although patients with asthma, they may not be more susceptible to contracting viral illness, and that is generally a fact compared with healthy controls or healthy individuals. They, however, do suffer a longer duration of symptoms and experience a more severe viral-induced respiratory illness. And I think this is important to note, is that they don't have a predilection to be more susceptible, but they may have a more extensive course of disease. So the important thing here to note is that viral infection and replication activate your innate and adaptive antiviral immune responses. And you have a pro-inflammatory cytokine response, you have chemokines produced and released, and you have the recruitment of inflammatory cells to the site of infection, which is the airway. The important thing to note is that influenza in particular and RSV, those 2 viruses actually cause cytopathic damage to airway epithelium versus the other viruses. So you're having a damaged epithelium that's now becoming more likely for other microbes to penetrate, and you're having more of an extensive inflammatory response, and this can lead to more severe disease. So, of note, the pro-inflammatory response propagates eosinophilia and stimulates the release of these cytokines, and this promotes asthma or atopic disease. I have some slides that will come further down just to illustrate this point clearly. With regards to viruses in the microbiome, I think what's important to note here is that rhinovirus, which is seen commonly throughout the year, this infection has been shown to alter your microbiome in the airway with increased detection of Streptococcus pneumoniae, Varicella, Catarrhalis, and Haemophilus influenza in children, both with and without asthma. Influenza also is associated with an increased risk of secondary bacterial infection. So, as much as you may get the viral infection, which is treated conservatively and from a symptomatic point of view, it's all very managed simply, you have the risk of developing bacterial infection. So again, viruses associated with recurrent wheezing and asthma. This table from UpToDate actually shows it quite nicely. Your major antigenic groups of rhinovirus are your A, B, C, your RSV, the coronaviruses that I mentioned, your human metapneumovirus, your parainfluenza. And Also, Bocovirus, I've mentioned there, can cause mild disease, but doesn't really exacerbate bronchiolitis or cause a wheezing phenomenon. Adenovirus, which is actually a DNA virus that can also cause upper airway disease. I haven't touched on this too much in this presentation because it's not seen commonly. This slide is an interplay between a high affinity receptor that is found on dendritic cells with the virus, and it shows what responses happen. So it's just a way to illustrate the interplay between the receptor and the virus. So here you will see, I can illustrate to you, you have a plasmacytoid dendritic cell, you have allergens that are around, and you have these high affinity receptors that are expressed by your dendritic cells. In a normal airway, your dendritic cells are generally the major sources of your type 1 and type 3 interferons, which actually respond to viral infections, and they actually will be cidal and decrease replication of virus and hence maintain a mild illness. So in a normal airway, that's what's going to happen. You have few receptors, you have dendritic cells that are releasing your interferon, and you have an uncomplicated event. But on this side, where you have an allergic airway with inflammation, when you have a virus that is introduced and you already have allergens in the presence, you have an increased expression of these high affinity receptors, which is on the FC region of your eosinophil. And what happens here is that you have an inverse relation between your expression of receptor and your interferon response. So you have many receptors, you have an increased viral replication because your dendritic cells actually decrease the amount of interferon that is released or released from the cell. And what happens is that you have increased viral replication, more severe illness, and with exacerbations of wheezing and an exacerbation of your pre-existing asthma. This slide just illustrates your rhinovirus and your respiratory syncytial virus in the way they attach to the receptors on your ciliated epithelial cell. To note the many different serotypes in your rhinovirus A, B, and C, as illustrated, more than 100 subtypes of this rhinovirus, and they tend to attach to your intercellular adhesion molecular 1 and your low-density lipoprotein receptors. Your RSV A and B, they tend to use your fusion protein to attach to the cell membrane, and they have a predilection for your pneumocytes as well. To note that your common viruses also can cause pediatric pneumonia, generally in those under 5 years of age, and I'm sure these are the ones that will be admitted to hospital and have a severe course of events. So I'm just going to go quickly through your respiratory syncytial virus, just to say that it causes mild disease generally in your children under the age of 5, also in adults, and most of the children by the time they're 2 years of age would have had an infection by RSV. It does not cause long-lasting protective immunity, and hence you have repeated infections. And as mentioned, it's effectively transmitted by large droplets. The RSV season in South Africa will differ from year to year, but generally we say it's from early Feb to mid-March is when it starts. Okay. As mentioned, the two major antigenic groups are your A and B, and the differences in the surface glycoproteins, which I showed you earlier, and we'll have another slide on that, is what actually speaks to their virulence and also speaks to how the vaccine that has now been developed relates to this. So you have different genotypes, and they can co-circulate within a season, and they can shift in dominance. Research published in the Journal of Infectious Diseases says that the RSVA genotype frequently replaces one another over successive seasons, while B tends to persist longer. And you will have regional differences in genotypes. Okay, just to note, we have an amazing surveillance system that the NICD actually runs, and they send out weekly reports. And if you visit their website and you click on any virus, it's amazing the amount of great literature that you will find there, from every single virus to bacterial pathogens as well. And I think it's great to visit the NICD site to get as much information as possible during our seasons. So here, just to note, over the past decade, excluding the time when COVID was prevalent, we found that your season, as I mentioned, typically begins in mid-February, which is around week 7, and the onset ranges from late January to late February, usually preceding the influenza season. So that's important to note. Again, the structure, your G and F protein. So there's your F protein and your G protein. These are key roles that allow for attachment. The G protein allows for attachment to your host epithelial cells, and your F protein causes membrane fusion. The RSVF is relatively well conserved, and it's highly similar between both your RSVA and B subtypes, but you can have small changes in your amino acids in sequences that have been studied. So your F glycoprotein or your fusion glycoprotein is the major target for neutralizing antibodies. And there's considerable debate as to whether your clinical severity is impacted by the subgroup of the infecting RSV strain. And in a review that I read published in 2024, the conclusion was that both subgroups cause severe disease, and none of the available evidence to date suggests any difference in clinical severity between the subgroups. As mentioned, risk factors for severe disease. And in children, we know over this period you do have quite a few hospital admissions from RSV. Generally, it's in those that are younger than 6 months, prem babies, those who have some chronic comorbidities, be it lung or heart disease, Down syndrome, if they're neuromuscular diseases. And these tend to be more at risk of severe infection and tend to have hospitalization. Generally, your self-limiting disease will just cause your cough and your rhinorrhea related to your upper respiratory tract, and that's for about a week. But when it spreads to your lower respiratory tract, then you would note, and especially the clinicians on the group will know, you're having all the different symptomatology and clinical expression of this, which takes the parent to the clinician and results in hospitalization. The important thing to note, I think, for me, is RSV and pregnancy, and I speak to this because this is a very specific group that tends to be at high risk of contracting respiratory syncytial virus, because children harbor this virus. It's relatively common amongst children, and pregnant individuals around young children at home will get this disease. As mentioned, for most adults, treatment is supportive and it causes mild disease. But now there is a new vaccine, a maternal RSV vaccination program that has come into play, and the vaccine is called Abrysvo, and it was launched by Pfizer, but officially came into play by the FDA in the US in August 2023. In the last month or so, I know this has been launched in our country, and it is now something that has been offered to our patients. It's the only RSV vaccine that has been approved in pregnancy. Of note, the prevention strategies, this is commonplace, but generally with RSV, as for any other virus, children should stay at home, okay, because they have high viral loads. They the ones that carry these viruses, and they promote spread of disease. So we will always strive to keep kids at home and try and practice good etiquette or educate them about good etiquette, which is quite difficult to do. But I think the important thing here to illustrate is that there is immunoprophylaxis for RSV in infants. And these monoclonal antibodies, palivizumab, and I'm not sure how easily available it is in the public and private sector, but this is one of the immunoprophylaxis that can be given to patients, to infants during the RSV season. And it has been shown to be effective in preventing severe infection. However, it's limited by the fact that it's very costly. You need to give it monthly. It's an intramuscular injection. And generally in countries like ours, accessibility may be a limitation and difficult. But in the US, the more recent one that has been launched in 2023 is nirsevimab. And this too targets the pre-fusion conformation of your RSV F glycoprotein, so the fusion glycoprotein. And the great thing about this one is that it has a long half-life and it has a very potent neutralizing effect on the virus. And you just need a dose of this, which gives you quite a long duration of protection for at least 5 months. And you give it at the beginning of the RSV season. So that I know is available overseas. I'm not sure whether it is available locally, but just a point on immunoprophylaxis, and previously the mention of Abrysvo for pregnant women. So of note, there are a good few RSV vaccines that have been approved by the US FDA. But here in South Africa, to date, Abrysvo is the bivalent vaccine that has been approved for pregnant women to prevent infection in the infant. So it can also be given to adults in greater, the age group of greater than 60 years. But I know this is something that's targeted for different countries, and in the US, that program is in play. So just a little bit about Abrysvo. So I think it's important to know that the goal of vaccination in the setting of pregnancy is actually to provide passive protection to the infants during the first few months of life, because that is when you get this disease and it can become a serious infection. And based on the available clinical trial data, the vaccine appears to be very safe during pregnancy. It reduces the risk of severe bronchiolitis in infants if maternal administration occurs at least 14 days between birth. So there's a stipulated time frame that you give it from 34 to 36 weeks. As with other inactivated vaccines, like your inactivated influenza vaccine, studies suggest that this vaccine can be effectively and safely co-administered with your diphtheria, your tetanus, and your acellular pertussis vaccine, which we also give in pregnancy. So just some information on the randomized trials that were conducted. The 6 trials looked at close to 18,000 pregnant patients and their infants, and they found that infants born to mothers who were vaccinated had fewer hospitalizations for lab-confirmed RSV disease within the first 180 days after birth compared with the placebo. And the rates of congenital abnormalities, your IUGR, and stillbirth were similar between your placebo and your active group. So this is good news. So that was RSV, and I think the important points there on RSV is prevention and also the fact that you have the ability now to protect pregnant mothers and their infants with the new vaccine that has been launched. Influenza viruses, we've known for many years pandemics and the epidemics that have occurred, but I thought just to visit a few points on influenza viruses. So just a quick overview. So the important thing to take away from this slide is that influenza A and B are the viruses that cause epidemics in humans, even though you may have different genera, but A and B are the important human viruses. The main antigenic determinants, again, are the surface proteins on viruses, and here on the influenza virus, we talk about the hemagglutinin and the neuraminidase transmembrane glycoproteins. These are critical for the pathogenesis and are the major targets for neutralizing antibodies for acquired immunity. You have 3 HA subtypes, H1, H2, and H3, and you have 2 neuraminidase subtypes, which is N1 and N2. And I'll explain this a little bit further on in the next slides. But these types or subtypes have circulated consistently in our population and are responsible for the annual epidemics. And this is what the viral watch and the surveillance looks at when we are actually targeting our vaccines and what needs to be tweaked in our vaccines through the seasons. So a pandemic can occur when you have a new and different influenza A virus that emerges and infects people. It has the ability to spread efficiently because you have no immunity to that. And that's an important thing to take away. So this is just an illustration again of your hemagglutinin and your surface glycoproteins, basically your hemagglutinin and your neuraminidase. There are 18 different subtypes, 11 different ones. It's just a different schematic. But I think here to touch on your drift and your shift. So how do flu viruses change? What happens in their genome? So we speak of antigenic drift and antigenic shift. Okay. So drift speaks to the small changes or mutations in the genes of your influenza viruses, and that leads to changes in your surface proteins that I mentioned. These changes associated with the antigenic drift happen continually over time. So they're continually happening as the flu virus replicates. The small, very small or minute genetic changes over time produce viruses that are closely related to one another because of their similar antigenic properties. So our immune systems generate antibodies to these surface proteins, and they usually are protective, they're neutralizing. So antibodies that your immune system creates against one flu virus will likely recognize and respond to a similar flu virus. So that we speak of is the cross protection. So antigenic drift is an important reason why we can get flu multiple times over the course of our lives. Okay, and as I mentioned, the composition of flu vaccine takes these important factors into account. The way we name influenza viruses is also quite interesting and a little bit complicated, but this is generally what has been accepted by the World Health Organization, defined in 1979. So the approach looks at the antigenic type, the host of origin, the geographical origin, what the strain number, and the year of collection. Right? So your antigenic type will generally be, example, your A, your B, your C, or your D. The host of origin, so for human origin viruses, there's no host that will be designated. But if you have a swine or an avian influenza, then it will be apportioned to where that host is or who that host is. So the geographical, the geographical origin will tell you in which site it was, whether, for example, Denver or Taiwan, and what strain number it is. So if you just follow that example, for example, your seasonal influenza A, which is the human example, is the H3N2, and that was in the year of collection, which would have been 2019, for example, and the strain number is 16, and the geographical origin is Perth. Okay. Okay, just to illustrate what you see on some of the reports. So I think here to take away a shift is a major change. It's an abrupt major change in the flu A virus, and you have a new hemagglutinin or both glycoproteins that are affected. So your body is going to see something, or your immune system is going to encounter something that it's never seen before. So this happens very infrequently, and I think the pandemics are what happens because of these major shifts. And we speak of the 2009 H1N1 pandemic. We speak about the 1968 pandemic with the H3N2 virus, and those are just listed there. This is just to illustrate the shift. Okay. I think it's important always, as much as influenza causes also self-limiting disease during the season, we should be cognizant of the risk groups where you have severe complicated influenza disease. And this would speak to the importance of vaccination, which I will discuss later. But this is a list from the 2025 guidelines, again from the NICD. It's a very comprehensive document, and I just pulled out some important pointers here. But the list is exhaustive. Again, treatment of influenza is something that is important, and I thought it would be important also to highlight the new drug that has come, so the new kid on the block, so to speak, the new drug that literally has come around after 20 years. So it's a new class of antiviral drug for influenza. The trade name is Xofluza. It is known as baloxavir marboxil. It generally targets the endonuclease function of the viral polymerase subunit. So it prevents the transcription of viral mRNA. And I think that's important. It's a different target site of how it works in comparison to your neuraminase inhibitor, which is your oseltamivir, or otherwise known as Tamiflu. So it allows for effective treatment of influenza or B virus infection, and it's just a single dose. So I myself have never taken one. I'm not sure whether any of the clinicians have prescribed it for severe hospitalized patients or those at high risk of mortality. So it will be interesting to know who's used this new drug. Going on to other treatment measures, I think it's important to know that generally amongst your hospitalized patients, you will treat them with an antiviral, with the appropriate antiviral, but also you will take cognizance of the fact that you can have secondary bacterial pathogens, so you will investigate appropriately. The atypical bacterial respiratory pathogens are also reported as part of our PCR multiplex testing, but I did not cover that in today's talk. I don't think with time constraints, I've left that out. Important to note as well that in ICU patients, there's a different type of PCR panel that they get exposed to, which is the pneumonia panel, which will also pick up bacterial pathogens as well as viruses. Antibiotics. So generally, antibiotics should not be used for the common cold or for any of your respiratory tract infections unless there's a high propensity in the certain type of patient where you want to give empiric antibiotics, but this is something that we don't routinely subscribe to. When there is the high risk of secondary bacterial infection due to those culprits, then we say your coamoxiclav, otherwise known as Augmentin, is a suitable empiric choice for those who are at risk for community-acquired pneumonia and who have a clinical indication. But it's a no-no to give antibiotics for viral infections because they will not work as an antiviral. Also, on the point of corticosteroids, these are not recommended for the sole indication of suspected or confirmed influenza or any other viral infection. And I think it's important to know that your randomized controlled trials that have been conducted for the use of corticosteroids said that it should not be used. There's no effect. I move on to influenza vaccine. And just to note here that vaccine must be given especially it should be given to everybody, but I think an important point is that in your high-risk populations and in that list that I showed you earlier, we should always target those as first prize. But generally, for all children greater than 6 months of age and pregnant women, we should advise vaccination for influenza. Egg protein allergy is not a contraindication to the influenza vaccine. I think that's an important one to note, especially with your concerned mothers. If you have egg protein allergy, you can still take the inactivated flu vaccine. Unless you have anaphylaxis, which is quite rare, then we shouldn't really have a problem in subscribing to taking the vaccine. So the formulation for 2025 covers your influenza A and your influenza B, and it's included in the quadrivalent vaccine. It's included, available in the private sector. And I think the important thing to note as well that there's ongoing research with regards to a universal influenza vaccine, where you can use it for cross protection. You can give it infrequently with long-lasting immunity. And currently, there are ongoing clinical trials, phase 3 trials, which are showing good effect. Here, they're looking at nanoparticles and adjuvants and viral-like particles, different modes of carrying the influenza epitope and keeping it stable against the immune system. And I think we basically need to watch that space. So hence what it will mean, the uptake will be good, less side effects, infrequent. So maybe you can take it 3 times in your lifetime and have good protection from it, and that would be great. I think it would be a miss for me not to say anything about the SARS-CoV-2, because there has been rumblings with regards to a new COVID variant. But this appeared in the NICD alert in June 2025. And just to take away from this, that there is a circulating variant of interest, and this is part of the Omicron descendant lineage. It was first detected in 2025 in Asia in January. So it is a variant under monitoring. In May this year, there were 22 countries that saw an increase in the so-called Nimbus variant. So the WHO said that the public health risk of this variant is low, and the current vaccines that we've all been exposed to are expected to provide protection against this Nimbus variant. And here in South Africa, and this was as of 10th of June, that variant has not been detected. But I think we need to know that, or take note of the fact that COVID testing or PCR testing is limited now. Not many people are truly doing PCR tests. It's more the antigen tests. So lab surveillance is not like it used to be before. So you get fewer specimens. But with that, we are still submitting for further sequencing, which is happening by the Network for Genomic Surveillance in South Africa. So surveillance for this is ongoing. I think if you need to know anything more about this, the NICD alert would be a great place to check. And they speak of this also in wastewater surveillance. So it's a good read. Moving over to our lab surveillance and statistics. So just to note again that you have a weekly surveillance update that the NICD sends out. And from December 2024 to July 2025, of their almost 4,500 samples tested, they found that 11% of cases were influenza, 16% of cases were RSV, and just under 3% of cases were SARS-CoV-2. And we also note a small incidence of pertussis. With AMPATH laboratory surveillance, we normally send this out, as routine to all our clients. And the important thing to note here is that over your epidemiological week 1 to 13, this pictogram with the bar graphs where you're seeing SARS-CoV-2 in blue that started up earlier in the year, and you see its decrease in prevalence, and you see your influenza A in the orange slowly starting to niggle and increase, but you see your RSV at around week 11, 12, and 13 increasing. So just comments on this is that looking specifically at influenza A and B, the number of specimens that were testing positive for influenza B remained low, but we found that there was an increase in your A positivity rate from 5.2% in week 9 to 8.6% in week 13. So hence, as we spoke, as the flu season came in, we saw your rise in influenza cases, and your RSV positivity rate testing also by week 13, up to 20% of specimens were positive. Looking at your multiplex PCR, and this picks up a whole of those common cold viruses that I spoke about, and I hope this projects clearly, but looking at your para-influenza, your rhinointro viruses here, I just want to illustrate the point of just how high in percentage in the age group 1 to 5, you'll see your rhinovirus in the month of February and March being around 60-62%. And also, I'm speaking to your RSV, which has increased to 23% in March, and your influenza is still low. Influenza A and B is still low, but influenza at 8.6%. Again, important findings during March from the AMPAT stats. In less than 1 year of age, the most common viruses were your rhinointroviruses and your RSV. In your group from 1 to 5 years, we saw that during the period of March, you had a preponderance of your rhinointrovirus, and your RSV definitely had escalated. Okay. When we looked at our April to May statistics on your multiplex PCR, again, just to illustrate the prevalence of rhinointro, but it certainly had decreased somewhat from its 60% in Feb-March to about 30% over April-May. And you still see your RSV being around just under 30% and your influenza at around 25%. Okay. So this just is looking at your epidemiological week, but relaying the same message. And if you look at the colors and you're just looking at the trends, if your eye follows the trends, you will see that your influenza A slowly increased over your weeks 11 through to 16, and then your RSV, which is the yellow also, the peak, and then the slow fall. So influenza actually went on later to week 22. Your RSV peaked between, say, at around week 16. Okay, it's the same thing. When we look at May and June statistics, then it's also showing you here that your influenza peaked in week 23 and your RSV positivity peaked at 23% at week 16. Okay, these are just illustrating similar points. So I think the takeaway there is that influenza and RSV and rhinovirus are your main circulating viruses. Yes, you will see your coronaviruses, your para-influenza viruses, but the ones that truly cause the problem and spread easily through our communities are the three major ones: rhinovirus, RSV, influenza. Just a quick point on viral diagnostics. I think it's just important to know that the current standard now for viral detection in most commercial laboratories is multiplex PCR testing. And at AMPAT, we have several different options for detecting respiratory pathogens. And really, the choice of test needs to be determined by the clinician based on their clinical assessment, what respiratory pathogen you are probably suspecting, how urgent is this result, what is the cost to the patient, the medical scheme, and you need to look at it in totality. The good thing is that you have a rapid turnaround time, and generally it has a favorable impact on the management of your patient. It's not routine. It's not something that we routinely recommend for uncomplicated illness, but it certainly has an impact on your treatment decisions in how you will manage your patients, whether you're going to be confirming the disease or not. And the fact that you have available treatment for influenza, for example, in severe cases, it would be good to know what the etiological virus is. So again, just to highlight here that our assays go through very vigorous quality assurance programs, and you can be rest assured that your result will be accurate. Your respiratory panels are highly sensitive and specific, so the PCR will tell you if it's positive, it's speaking to what's being picked up, so the sensitivity is great. So the respiratory panel PCR is your comprehensive panel that tends to be used for inpatient use, where your co-tri PCR tends to be for just your influenza, RSV, and your SARS-CoV-2. So this tends to be used at an outpatient level, and medical aid does reimburse for inpatient for your respiratory PCR and your co-tri PCR as well at outpatient level. Again, just to note that you have different patient populations that you would test, but generally in your high-risk pediatric patients, you would want to test for this on either a nasopharyngeal swab or an aspirate. Again, in your immunocompromised patients and your high-risk groups. This is just to illustrate what the panel looks like. And I think just to end from here, because we really are out of time now, is that the point of this talk today was not to tell you too much about the heaviness of a discussion around viruses, but just to tell you what prevails, what there are the important points of some of the common circulating viruses, and also to illustrate that most of the time it does cause mild self-limiting conditions, but we must emphasize that there are vaccines available for influenza and for RSV, and they are the primary means for preventing seasonal influenza infection. So we must promote that in our practices. Given early would be first prize, so you must give it sufficiently early, your influenza vaccine, so that your antibody response, has sufficient time to respond. But you can actually take it at any time because it would speak to herd immunity. So actually, your community will be protective and you will have less transmission of virus. The RSV vaccine, I think that it is now available, and in our pregnant population, we should ensure uptake, and we should drive this because it has good impact as evidenced by the literature. The point on diagnostic testing, certainly make that as an individual decision at the level of your patient and use it judiciously, and understand that your treatment decision should be based on your clinical presentations and should not be delayed based on pending lab information. I thank you for your time.